Okay, so today is our first class MCQ discussion, full class, and physiology. Today we will discuss about physiology MCQs. So the first MCQ you can see is written: electrogenic sodium potassium ATPase plays a crucial role in cellular physiology by using energy in ATP to extrude 3Na out of cells in exchange for taking 2 potassiums into the cells, and using energy in ATP to extrude 3 potassium out of cells in taking for sodium into the cells, and using the energy in moving sodium into the cell or potassium outside the cell to make ATP, and using energy in moving sodium outside the cell or potassium inside the cell to make ATP. So the answer will be number A, using energy in ATP to extrude, this is the point, to extrude 3Na out of the cells in exchange for taking 2 potassiums. It takes 2 potassiums into the cells and 3 sodium out of the cells. So sodium potassium ATPase catalyze this hydrolysis of ATP to adenosine diphosphate from triphosphate to diphosphate, and while converting this from tri to di, it gives energy which is used to extrude 3Na, 3 sodium ion from the cells, and it takes 2 potassium ions into the cell. For each molecule of ATP is hydrolyzed. So this activity also, remember, this activity is inhibited by Waban and related and related digital and also digitalis glycosides, which are used for so this activity is inhibited by Waban and digitalis glycosides, which are used for CHF, congestive heart failure. Active transport of sodium and potassium is a major energy consuming process in the body, and in body it accounts for 24% of energy utilization by cells and 70% of energy utilized by neurons. So anyway, the main thing is that 3 sodium ion will be out of the cell. 3 sodium ion will be out of the cell. Instead, it will take 2 potassium ion. This is very important point. Okay, next. All of the following are calcium binding protein, except, that means which one of the following is not calcium binding protein out of troponin I, calmodulin, dystrophin, calcineurin? And the answer will be dystrophin. Dystrophin is actually, it's a structural protein. Dystrophin is a structural protein which is situated in subcercolemmal region of the muscle fiber membrane. And other protein you see, these are all calcium binding proteins. Troponin is involved in the contraction of skeletal muscles and cardiac muscles. And then we got calmodulin, helps in phosphorylation of myosin and muscle contraction. Cal binding, vitamin D dependent calcium binding proteins, and calcineurin, option number D, calmodulin activated protein which inactivates calcium ion and plays a role in activating the T cells. And another thing to be remembered is immunosuppressive drugs can inhibit calcineurin. Okay. So next question, which of the following is the correct statement? Desmosome help to attach these cells to basal lamina. Hemidesmosome help to attach cells to each other. Tight junction permit the passage of some solutes between cells. And gap junction prevent the movement of proteins in the plane of movement. So the answer is tight junction permits passage of some solutes between the cells. Okay. So the function of tight junction, always remember, it allows the passage of some ions and solutes in between the adjacent cells, and that is called paracellular pathway. and it prevents movement of proteins in the plane of the membrane, but it allows the passage of ions and solutes between adjacent cells. So that is to be remembered. Important points. Otherwise, here you can see some structures of tight junctions, gap junctions, and other anchoring junctions like desmosome, hemidesmosome. Okay. Okay, so next MCQ, all are true about the body fluids and electrolytes except. So which one of these is not true? Intracellular fluid constitutes 40% of body weight. Interstitial fluid constitutes 15% of body weight. Insensible losses are 400 to 800 ml per day, and chloride inside cells is more than outside the cells. Okay, so the answer will be number D. That is, chloride inside cells is more than outside cells. So very important point. Another important point is concentration of ions inside the cell and outside the cell. Chloride ions, it's 9, 125. Just remember inside, outside, 9, 125. Potassium, 150, 5.5. Sodium, 15, 150. Okay. So these are the three ions that are that can come in FMG also. So and the concentration of each of these ion inside the cell and outside the cells. Motor neuron of spinal cord is unipolar cell, bipolar cells, pseudo-unipolar cells, multipolar cells. So motor neuron of spinal cord is unipolar cell, bipolar, pseudo-unipolar, multipolar. So the answer is multipolar. This is quite information based direct question. Unipolar cells are invertebrate cells. Bipolar cells are bipolar cells of retina. Pseudo-unipolar cells, example is ganglion cells of dorsal root, while multipolar cells are motor neuron of spinal cord, which is the question, motor neuron of spinal cord. Then we have pyramidal cells of hippocampus, Purkinje cell of cerebellum. So these three are multipolar cells. These three are multipolar cells. Okay. So next question, all are true about cardiac muscle except. All are true about cardiac muscle except phase 1 of depolarization is due to opening of voltage gated sodium channels. Phase 2 of depolarization is due to slow opening of voltage gated calcium channels. Phase 3 of repolarization is due to closure of calcium channels, while tetany of cardiac muscle is never seen. Number D. So A, phase 1 of depolarization due to opening of voltage gated sodium channel, due to slow opening of voltage gated calcium channel, due to closure of calcium channel, and number D is tetany of cardiac muscle is never seen. So the answer will be number A, that is phase 1 of depolarization is due to opening of voltage gated sodium channels. So what are the phases of cardiac muscle? You see the phases of cardiac muscle. Phase 0 is rapid depolarization due to opening of voltage gated or voltage sensitive sodium channels. Phase 1 is rapid repolarization. So first depolarization, then repolarization due to closure of the sodium channels and opening of one type of potassium channels. Phase 2 is plateau phase due to slow and prolonged opening of the gated calcium channels. Phase 3 and 4, final repolarization to resting membrane potential and is to closure of calcium channels and slow delayed increase of potassium efflux. Increase of potassium efflux starts from phase 4. And phase 3 and 4 is repolarization and final repolarization to RMP, resting membrane potential. And phase 2 is the plateau phase. In fact, the question also can come like this: which phase is what type of phase? Like rapid depolarization phase 0, rapid depolarization phase 0, rapid repolarization, phase 1, plateau phase, phase 2, final repolarization to RMP, phase 3 and phase 4. Okay, so these names are also important. Okay, another thing is that cardiac muscle remain in refractory period till phase 4, and there is no tetanic contraction in skeletal muscles in cardiac muscles. Okay. There is no tetanic contraction in cardiac muscle, but it can occur in skeletal muscle. So tetanic contraction occurs in skeletal muscle, but no tetany in cardiac muscle. Okay, that is to be remembered. So the option D, tetany of cardiac muscle is never seen, is correct. That's why we have not choose that option, because the question is all are true about cardiac muscle except which one of the following is not true. And you see phase 1 of depolarization, it should be rapid repolarization, then it is true. So this is wrong actually. The fact is wrong. That's why this is the answer. Anyway, next question. Which of the following is responsible for skeletal muscle relaxation? Dystrophin, tropomyosin, myosin, actin. Dystrophin, tropomyosin, myosin, actin. Which of the following is responsible? So option answer will be tropomyosin, right? So tropomyosin, what is its function? Its function is it acts as relaxing protein at rest by covering the sites where myosin binds to actin. And dystrophin is sarcolemmal protein providing calcium input to sarcoplasmic reticulum. Calcium ion is useful in coupling the actin myosin complex. Okay, so what happens in contraction and relaxation of skeletal muscle? First of all, we'll revise a little bit. Myosin heads hydrolyze ATP. Myosin head hydrolyze ATP and become reoriented and energized. Okay, so myosin heads hydrolyze ATP and become reoriented and energized. Number 2, what is happening? Myosin heads bind to actin, myosin and actin, right? Myosin binds with actin, forming the cross bridge. This is very important point, very important phase of contraction and relaxation mechanism. They form the cross bridge. So first of all, they were not forming the cross bridge. There was gap, and now they form the cross bridge. Then number 3, myosin cross bridge rotate. Then it start rotating. Then you see this start rotating. The angle becomes more steep like this. It was first like this. Okay, so the myosin cross bridge rotate towards the center of sarcomere. That is called power stroke. And lastly, as myosin heads bind with ATP, the cross bridge detach from the actin. Okay, so this is the contraction and relaxation of skeletal muscle in short. So always remember the relaxation is done by tropomyosin. And actin and myosin both are involved in contraction and forming the cross bridge. So next, which of the following is a unique feature of smooth muscle? Unique feature of smooth muscle: tetany, plasticity, excitation, contraction, coupling, and not recall. Which of the following is a unique feature of? Okay, the answer will be, plasticity. Okay, so unique feature of smooth muscle, that is smooth muscle has 20% of myosin content and 100-fold difference in ATP use when compared to skeletal muscle. But it can similarly force, but it has similar force per cross-sectional area, and special feature of skeletal muscle The special feature of skeletal muscle is, however, plasticity, where it behaves like viscous mass rather than rigid structured tissue. So an example of plasticity is that urinary bladder tension rises very slowly when urine amount is minimal. But when the amount of urine approximately about 150, bladder can contract forcefully. Okay, this is not skeletal muscle, this is smooth muscle. The special feature of smooth muscle. Smooth muscle is the plasticity which the skeletal muscle doesn't have. And the smooth muscle can, what is this plasticity feature? This is the feature that urinary bladder tension rises very slowly when urine amount is minimal. But when the urine amount approximate about 150 ml, the bladder can contract fully. So the thing is, stimulation is the how much the bladder is fulfilled. If the bladder is not filled completely also, then also the bladder can contract forcefully. That is called plasticity. That smooth muscle has this feature, which skeletal muscle doesn't have. Okay, this is about plasticities, smooth muscles, unique feature. So here is a difference between three types of muscles given. You see the locations mostly, then appearance, cell shape, nucleus, striated muscles like skeletal and cardiac muscles are striated, but not smooth. And not smooth muscle. Smooth muscle has no striations. Then the control, skeletal muscle are only voluntary, but cardiac and smooth are not. They are involuntary. And the skeletal muscle help the body to move, whole body to move. But cardiac muscle, as you know, it is present only in heart. So it helps in only in propelling the blood throughout the body. And in smooth muscle, for smooth muscle, it compresses the organs, ducts, tubes, okay, like urinary bladder, all these things. So this is about difference of three muscles. Now, next question, all are secondary messengers except. All are secondary messenger except number D, guanylyl cyclase. Guanylyl cyclase is an enzyme that catalyzes the formation of cGMP. So it is important for vision in both rods and cones present in the eye, right? Rods and cones. And second messengers bring about, what are second messengers? They bring about the short-term changes by altering the enzyme functions, triggering exocytosis, and also lead to alteration of transcription of various genes. Okay, so examples of second messenger: CMP produced by action of adenyl cyclase enzyme and stimulates protein kinase A, which catalyze the phosphorylation of the proteins and altering their activity. And for example, like cholera toxin increases CMP levels, and various pertussis toxin inhibit it. And IP3 inositol phosphate diffuses into endoplasmic reticulum and triggers calcium release. And DAG is diacylglycerol walks to IP3. So these are three secondary messengers or second messengers. But guanylyl cyclase is an enzyme that catalyzes the formation of cGMP, and which is important for vision. Okay, next, transport of sodium with sugar from enterocyte is example of transport of sodium with sugar. Transfer of sodium with sugar from enterocyte is an example of diffusion, active transport, facilitated diffusion, and secondary active transport. Obviously, secondary active transport. Okay, secondary active transport. As we know, secondary active transport, what happens? The ATP-driven sodium and potassium pump stores energy by creating a steep concentration gradient for sodium entry into the cell. And as sodium diffuses back across the membrane through a membrane co-transporter protein, it drives glucose against its concentration gradient into the cell. Okay, so these are the two steps of secondary active transport. And cell transport, just to revise the cell transport, there are two types of cell transport. One is active and another is passive. Active means simply ATP is used. ATP is used. And in passive, there are two types. One is diffusion, another is osmosis. In diffusion, there can be simple diffusion, there can be facilitated diffusion, and osmosis means it's passive flow of solvent. It depends on the number of solid particles. Okay. And another thing is sodium potassium pump. Always remember, we have done it already, that it pumps out 3 sodium ion from the cell, and it pumps in and for that it pumps in 2K plus inside the cell. It maintains the cell volume, also helps in the maintaining of cell volume. Next, permeability of which of the following ion leads to RMP? Permeability of which of the following ion leads to RMP? Chloride, potassium, sodium, calcium. So the answer is potassium. Potassium permeability leads to RMP. So what is RMP? RMP is the resting membrane potential, right? Resting membrane potential, and this is resting membrane potential is the potential difference across the cell membrane in millivolts, which is conventionally we use 70 millivolt, right? 70 minus 70 millivolts. And it is established by diffusion potential that results from concentration difference of permeable ions. Another thing is that sodium ion is actively transported out of neurons and other cells, and potassium is actively transported into the cell. Potassium is actively transported into the cells, but because potassium permeability at rest is greater than sodium permeability, potassium channels maintain the RMP. Okay, this is the reason why potassium ion can maintain the RMP, or resting membrane potential. So remember, resting membrane potential is maintained by potassium ion. Neuropraxia. True about neuropraxia is it is neuronal degeneration. No, this is true about disruption of conduction. Disrupted axonal sheath, structural change of nerves. So the answer will be disruption of conduction. So it refers to physiological disruption of conduction only. There will be no anatomical abnormality. Always remember, in neuropraxia, no anatomical abnormality or disruption. Okay, only physiological disruption of conduction. Of what? Of conduction. Conduction disruption actually. Both axons and myelin sheath are intact. No structural change or degeneration occurs in case of neuropraxia. Neuronal degeneration is seen in if occurs, is seen in crash nerve injury, fetal development, senescence means senility, old age. And axontemesis means axon and myelin sheath disrupted, but endoneural perineurium and epineurium sheath is intact. Okay, neural degeneration is present. In axontemesis. And neurontemesis is complete division of the nerve. The term refers to complete division of the nerve. Axon neural tube are divided, like either perineurium, epineurium, endoneural sheath are divided. Neural degeneration is present. Neural degeneration is present. So neural degeneration is present both in neurontemesis and axontemesis. Okay. Next is all of the following muscle bands are covered by actin filament. Covered by actin filament except A, Z, I, and H. Very important question. So except. So all of the following muscle bands covered by actin filament. So muscle bands that are covered by actin filaments are A bands. Yes, A bands covers the actin filament. Z band covers the actin filament. I band also covers the actin filament, but not H band. So H band will be the answer. H band will be the answer. Now we will revise all these bands. First of all, this is a picture of what? This picture has come as IBQ in the very recent FMGE also, 2018 or 19. It has come. We need to revise it. This is the image of sarcomere, which is a functional unit of myofibril, which is divided into several bands. First of all, you see the area between two Z lines. This is one Z line, another is this Z line. Okay, this area between two Z line is called sarcomere. This is called sarcomere. This is sarcomere. And the thick filaments, which are about the diameter of the thin filaments that are made up of myosin. The thin filaments are made up of actin, tropomyosin, troponin. So the thick filament, which are about the diameter of the thin filaments, are made up of myosin, and the thin filaments are made up of actin, tropomyosin, and troponin. Okay. So next we got H band, lighter H band. This is the H band. This and this, and in the middle of the H band, that is M band. Lighter H band in the center of A band. In the center of A band. A band is from here to here. No, sorry, A band is from here to here. This is A band. This is A band. This is A band, and in the center of A band is M line. You see this black colored M line. This is M line. And this white color around the M line is H band. This is H band. Okay. And these Z lines, this is Z lines, they transect the fibrils and connect to the thin filaments. If a transverse section through a band is examined under the electron microscope, then each thick filament is seen to be surrounded by 6 thin filaments in a regular hexagonal pattern. So all these bands should be remembered. Which are the I mean, which is Z band, which is I band is this. This is I band, H band, A band, M line. M line is between A band, and two Z lines adjacent to the area between the two adjacent Z line is sarcomere. That is, this is also very important MCQ. Very important MCQ. So they can give you like sarcomere is area between these two Z line, A band, then the area between the area of I band. So in this way they can give, and consecutive Z line. Consecutive Z line, yeah, area between consecutive Z line. So remember which one is the sarcomere, which is M line, and which is I band, A band, H band. Okay, they will not give this labeling in the exam. Like this they will give without any labeling. Okay, next is macula densa is responsible for densa is responsible for tubular feedback. Tubular feedback, renal reflex, maintaining filtration fraction. Okay, so macula densa is responsible for tubular feedback, tubular feedback, renal reflex, and maintaining filtration fraction. So the answer will be tubular feedback. Now, what is tubular? What it does? It maintains the constancy of load delivery to distal tube or distal convoluted tubule (DCT), and the sensor for the response is macula densa. Macula densa controls the flow. Macula densa controls the flow of tubular feedback. Tubular feedback is decrease in GFR occurs, and it promotes what? Solute reabsorption in PCT. If GFR decreases, glomerular filtration rate decreases, it leads to promotion of solute reabsorption. Solute reabsorption in PCT. In PCT, and solute reabsorption in thick ascending limb of Henle also. Henle's loop, ascending loop of Henle or limb of Henle, thick limb of Henle. Plus, so PCT plus thick Henle's limb. Okay. Solute reabsorption occurs, and renal is increase in ureteral pressure in one kidney. Increase in ureteral pressure in one kidney leading to decrease in efferent nerve activity to contralateral kidney. That means if one kidney experience increase in pressure of ureter, then it leads to decrease in efferent nerve activity to contralateral kidney, permitting an increase in excretion of sodium and water. That is renal reflex. And filtration fraction is the ratio of GFR to renal plasma flow. It's a ratio of GFR to renal plasma flow with a normal fraction. Normal value is 0.16 to 0.2. In hypotension, what happens? GFR is affected lesser than renal flow because of efferent arteriolar constriction. So another thing we will revise is macula densa. So macula densa, what is macula densa? Macula densa are JGA apparatus cells of JGA, juxtaglomerular apparatus. Juxtaglomerular apparatus. Glomerular apparatus. Okay. So these are the cells. This is one of the cell, one type of cell of JGA. It releases adenosine, causes vasoconstriction. Macula densa. Actually, when macula densa comes into play, if blood pressure is very much low, it releases adenosine. BP low, so it releases adenosine and cause vasoconstriction of the efferent arteriole and will slow the GFR. It slow the GFR. So if GFR gets slow, then what will happen? water excretion will be delayed. So water will be more conserved in our cardiovascular, in our vascular system. Water will be conserved. So water loss reduces the frequency or the amount of water loss from vessels, and blood pressure will be maintained. BP will be maintained or increased. Maintained, at least maintained to increase a little. Okay. So this is the main function of macula densa. Macula densa is present in here. You see, this is afferent. This is afferent arteriole. This is afferent arteriole, and all the cells of JGA are present here. And macula densa are this. This is macula densa. Okay. Other than macula densa, we have another modified smooth muscle cells of afferent arteriole with epithelioid appearance. It secretes renin. And it takes part in these cells. These cells secretes renin. It takes part in RAS, renin angiotensin aldosterone system. RAS, what it increases BP, right? Increases BP. How? By reabsorption of sodium and water. And Lassis cells, we have another type of cell called Lassis cells or mesangial cell. Lassis or mesangial cell. Lassis cells or mesangial cells. This is the Lassis cells. So this is macula densa. This is macula densa. This is Lassis, number 1 macula densa, Lassis, and renin secreting granular cells. Next question, CO2 is mainly transported in blood in which form? Dissolved SCO3, carbamino, and gas form. So very good question. Answer will be SCO3. Okay, CO2 is transported. So mainly CO2 is transported in blood in 3 forms. That is bicarbonate, mainly transported. The answer, this bicarbonate, 70%. About 70% CO2 is transported in this form. And so when CO2 diffuses into RBC, this will be 2. When CO2 diffuses into RBC, it reacts chemically with water. And with the help of enzyme, carbonic anhydrase, it's converted to SCO3 plus H plus, SCO3 minus plus H plus actually. SCO3 minus plus H plus. As dissolved CO2, in dissolved form 6 to 7% only. 6 to 7%. And as carbamino compound of hemoglobin, other plasma protein 20%. This is 20%. So 70, 80, 90, and almost 97. So almost done. So the question is mainly CO2 is transported as bicarbonate form. So next, osmotic diuretics. Diuretics acts on osmotic diuretics acts on PCT. PCT means proximal convoluted tubule. Proximal convoluted tubule, right? This is proximal convoluted tubule. So what are the drugs? First of all, we will revise the available diuretics. We have one is osmotic, loop, thiazide, potassium sparing, CAI or carbonic anhydrase inhibitor. So osmotic diuretics, they inhibit water and solute reabsorption. Okay, and loop diuretics inhibit sodium, potassium 2Cl minus co-transport. Thiazide diuretics inhibit sodium and chloride co-transportation. And potassium sparing diuretics decrease sodium reabsorption and potassium secretion. Decrease sodium reabsorption and decrease potassium secretion. And carbonic anhydrase inhibitor inhibit H plus secretion and SCO3 reabsorption which reduces sodium reabsorption. So in simple word, diuretics, what it does, it increases water excretion. Okay, it increases water excretion. That's why sometimes it is also given in hypertensions. So water excretion, that means how water excretion can be increased. Two methods. Either you inhibit water reabsorption. You know that in nephron the urine formation takes place. This is the nephron, right? And this in these tubules, this is PCT, proximal convoluted tubule. Then we have descending limb of Henle. Then we have ascending limb of Henle. Then distal convoluted tubules and collecting duct. So if we can, some substances are secreted into the tubule and some substances are reabsorbed from the tubule. So whatever substances or the amount of substances that will be secreted into the tubule will be excreted via urine, through urine. Okay, in this way the kidney balances the amount of substance also by secretion and reabsorption. Okay, so if we inhibit the nephron to reabsorb water, then this means what? This means there will be excretion of water. So inhibition of water reabsorption is one of the very important and main mechanism of action of this diuretics. Okay, and also sodium, water excretion and also sodium excretion. It like this. So mannitol is the osmotic diuretic. It inhibits water and solute reabsorption. And loop diuretic, it inhibits the co-transportation of sodium potassium with 2Cl. Inhibit the co-transport. What happens is that by this co-transportation, 2Cl minus is being secreted and sodium potassium. So 2Cl minus is secreted, sodium potassium is taken in. That means reabsorb. This reabsorption is inhibited. Thus, sodium is excreted only through the urine. Okay, and thiazide diuretic inhibits sodium chloride co-transport. Potassium sparing diuretic decrease sodium reabsorption and decrease potassium secretion. Decrease sodium reabsorption. So so that means potassium sparing diuretic, what it does, it allows sodium to be present inside the tubule, so that it can get excreted by the urine. Okay, and carbonic anhydrase inhibit H plus secretion and HCO3 reabsorption. Ultimately reduces the reabsorption of sodium, so letting the sodium inside tubule in order to excrete the sodium ion. Okay, now see which diuretic acts on which site of the nephron. That is very important. Just remember by the site, like first PCT comes. PCT in CAI means carbonic anhydrase inhibitor and osmotic, that is mannitol, osmotic diuretic acts on PCT. Then ascending limb of Henle, loop diuretics. Loop. In DCT, distal convoluted tubule, thiazide. And collecting duct. In collecting duct, potassium sparing or potassium spironolactone. So also remember the names of the drugs, examples of drug like loop diuretics includes furosemide, torsemide, thiazide diuretic, hydrochlorothiazide, benzthiazide, chlorthalidone, indapamide, potassium sparing diuretic, spironolactone, amiloride, acetozolamide is CAI, carbonic anhydrase inhibitor. Okay. Okay. Okay, next is true about RAS. True about RAS. True about RAS is aldosterone is made by hypothalamus. Angiotensin 2 causes arteries to constrict. Aldosterone 2 secrete more sodium and water. Blood pressure is maintained in RAS. So the answer will be angiotensin 2 causes arteries to constrict. It constricts arteries, it increases BP. Because another thing is missing actually, constrict arteries decrease GFR or prevents, or we can say prevents increase of GFR, glomerular filtration rate, and thus water and sodium conserved in vascular system. So it increases BP and it increases BP in this way. Now this is the full revision of renin-angiotensin-aldosterone system. It ultimately increases blood volume and pressure. So in hypotension, it acts. Okay, RAS system. When pressure drops, then RAS system acts and maintains blood volume and maintains the pressure. Or we can say if hypotension has occurred, then it again comes back by RAS system. So first of all, angiotensin in the presence of renin. Renin is secreted by, we have studied already, renin is secreted by juxtaglomerular cells. This renin secreting cell, juxtaglomerular cells. Okay, renin secreted by these cells. So renin, in the presence of renin, angiotensin becomes angiotensin 1, and in the presence of S, this is angiotensin converting enzyme. Angiotensin converting enzyme secreted from lungs. Okay, enzyme. Okay, in the presence of S, it becomes angiotensin 2. Now, direct effect. It has direct effect. Number one, it causes arteries to constrict, increases cardiac output, resulting in an increase in blood pressure and volume. So what happens? Why cardiac output increases? Because arteries constriction of arteries occurs, and if constriction of arteries occur, there will be more flow of blood, and cardiac output will increase. And decreases glomerular filtration rate. Just we discussed it in the previous slide. It decreases glomerular filtration rate. And resulting in water retention. It increases thirst. Angiotensin 2 increases thirst, so that we take more water. These are all direct effects. Another other effects. It encourages the secretion of aldosterone and ADH. So aldosterone means aldosterone will what it do? It causes nephron. It causes DCT to reabsorb more sodium and water. Aldosterone. So we call it renin, angiotensin, aldosterone system. So this aldosterone will reabsorb more sodium and water, which increases blood volume. Okay, and it works in aldosterone acts on DCT. Very important. All these points are also very important for physiology for FMG. Okay, so aldosterone acts on DCT. Aldosterone acts on hair. Also remember aldosterone acting site is distal convoluted tubule here. Okay, and ADH, and as you know, anti-diuretic hormone, right? Anti-diuretic hormone. It mediates insertion of aquaporins into nephron collecting duct cells. As a result, more water is reabsorbed into the blood. And it increases sodium reabsorption also in the middle of the kidney. So antidiuretic hormone, as the name suggests, is just opposite to diuresis. It is opposite to diuresis. In fact, the whole RAS system, it is opposite to diuresis. It increases amount of water and sodium. It conserves water and sodium into the circulatory system. Okay, and also remember where ADH is secreted. ADH is made in the hypothalamus, released by posterior pituitary. These are all important points for physiology. So made in hypothalamus, released by posterior pituitary, is about ADH. Then we have ANP, atrial natriuretic peptide, which is made by atrial cells in the heart, and renin, produced by the kidney, takes part in RAS system, or RAS, renin angiotensin aldosterone system, and converts angiotensin to angiotensin 1. Then angiotensin is made by liver, and aldosterone is produced by the adrenal gland. So angiotensin 2 triggers the secretion of aldosterone. So it is produced in where? Adrenal gland. Remember, produced in adrenal glands located on the top of kidney. So this is about RAS, renin-angiotensin aldosterone system. Okay. Next question from GIT. All of the following are false about gastric juice secretion, except false about gastric juice secretion, except false. So which one of the following is true about gastric juice secretion? Which of the following is true? 20% in gastric phase. Anyway, the question is a little wrong. It should be all of the following are true about gastrics. True, it should be true about gastric juice secretion, except 20% in the gastric phase. This is wrong. This should be 70%. Gastric phase is 70%. Okay, 70% of secretion, and cephalic phase accounts for 20% of secretion. Intestinal phase also 10%. It is written 20 approximate. Okay. So gastric phase is largest one. 70% is of gastric juice secretion accounts for gastric phase. This is very important. So this fact is wrong. That is why this is the answer. This is the answer. And the rest of the options, 20% in cephalic phase is true. 20% in intestinal phase is also true. It's mentioned 10%. So approximate 20% we can consider it. 70% is also true. Okay. So cephalic phase, it occurs when the food is not even in the mouth. It's not even in the mouth. It is stimulated by sight or the smell of food. So that time only gastric acid secretion starts. Gastric acid secretion starts just by sight and smell. Just by sight and smell. The impulse in cortex and reach the gland by dorsal motor nuclei and through vagus nerve. Vagus nerve is involved and dorsal motor nuclei is involved. Always remember this. Vagus nerve controls secretion of gastric acid. Secretion of gastric acid. Okay, next is gastric phase, when the food reaches the mouth and occurs by vasogastric reflex and enter enteroreflexes by gastrin. Okay, so when the food is in the stomach, that up to from the stomach to the from the mouth to the stomach. Okay, it is gastric phase, which is the stomach. Gastric phase. And intestinal phase, as the name suggests, when the food, especially in the duodenum, it causes reflex feedback. And 10% gastric acid is secreted at that time. So most of the gastric acid is secreted when the food reaches the stomach. So when the food reaches the stomach, food in stomach means what? Food in stomach means gastric phase. Gastric phase. So these are the phases of HCl secretion and gastric juice secretion, other enzymes also, HCl plus other enzyme secretion. These are the phases of gastric secretions. Okay, so number one is cephalic phase, when we just see the food and take the smell. Okay, smell the food, then 20% of the secretion occurs. When it reaches the stomach, then 70% secretion occurs. And when it reaches intestine, then also there will be secretion of gastric juice. That will be accounting for 10 to 20%. That will be accounting for 10 to 20%. Okay, so this is cephalic phase, this is gastric phase, and this is intestinal phase. Next, which one of the following is false statement? Which one of the following is false statement? Principal glands are located in body and fundus. Parietal or oxyntic cells are source of gastric acid and intrinsic factor. Parietal and chief cells predominate in cardiac site of stomach. Chief cells are the source of pepsinogen. So which one of the following is false? False is parietal and chief cell. Parietal cells and chief cells predominate in cardiac of the stomach. Okay, this is false. Why? Okay, we will go option by option. First of all, principal glands are located in body and fundus. Okay, so principal glands are located in body and fundus. It is true. This is true. Okay, parietal or oxyntic cell are the source of gastric acid and IF, intrinsic factor. This is also true. Parietal and chief cells predominate in the stomach cardiac site, cardiac of the stomach. Parietal and chief cells. No, that is not true, because the parietal and chief cells, they comes under principal glands. Actually, they comes under principal glands. So they also should be located at the body and fundus, not at the cardiac. Okay, they comes under principal glands. And chief cells are the source of pepsinogen. Chief cells are the source of pepsinogen. That is also true. Along with pepsinogen, chief cells are also the source of renin. This is not that renin. That renin is secreted by kidney. Okay, this is digestive enzyme. Renin, lipase, and they also contain zymogen containing abundant RNA and intensely basophilic. So chief cell. So this is about chief cell. Okay, and other glands present are gastric glands divided. Other glands are pyloric glands. Sorry, pyloric glands are mostly populated with mucus secreting cells. These are mucus secreting cells. Populated with mucus secreting cells. Parietal cells are very few and chief cells scarce. Okay, so this is about pyloric gland. So these all are gastric glands. These all are gastric glands. Gastric glands can be divided into three groups: pyloric, principal, and cardiac. Cardiac glands are mucus secreting cells, predominantly mucus secreting cells. Mucus secreting, predominantly. And pyloric are also populated with mucus secreting mainly, and but parietal cells are few and chief cells are also there, but very little amount. Okay, so these are the three group of glands present as gastric glands. Okay, next question. Starch in small intestine is digested by. Okay, starch in small intestine is digested by pancreatic amylase, tyalin, lipase, mucin. Okay, so pancreatic amylase, tyalin, lipase, mucin. So answer will be pancreatic amylase. Pancreatic amylase. So we will go in details. So this MCQ is little tricky because mainly the starch is digested by tyalin only. Okay, but in intestine, pancreatic amylase. This is in intestine. So saliva contains two major types of protein secretion. Number one is serous secretion and another is mucus. Okay, and tyalin, you know, is a type of is a serous secretion of saliva, and saliva pH range is 6 to 7. And activity of salivary amylase is blocked by acid of the gastric secretion due to low pH. And another thing is that before food and its accompanying saliva become completely mixed with gastric secretion, as much as 30 to 40% of the starches will be hydrolyzed mainly to form maltose. So when we give food to mouth, we introduce the food into mouth, saliva starts working, and until the food reaches the stomach, saliva works, and while working, saliva hydrolyze 30 to 40% of starch. 30 to 40% of starch, and the rest of the starch will be digested in small intestine, not by saliva. Because when food reaches the stomach, food means, always remember, food plus saliva mixed. Bolus is called bolus. Food plus saliva. So when it reaches the stomach, what it does, what happens? Acid, gastric acid inhibits saliva. So saliva becomes useless at that point at the in stomach. So obviously from stomach, saliva cannot work anymore. Okay, so what will happen to the starch? What will happen to this food? Means mainly we are talking about starch here. What will happen to starch? So starch will then go into intestine. And in intestine, in the intestine, the rest of the starch digestion will take place by another enzyme, that is pancreatic amylase, not salivary amylase, rather pancreatic amylase. Okay, so remember, starch takes a little time to digest. That's why. Okay, saliva starts digestion of the starch, but up to stomach it can digest about 30 to 40%, but rather and rest of the starch is digested in intestine, small intestine, by pancreatic amylase. Chymotrypsinogen is activated into chymotrypsin by trypsin. So the question was, chymotrypsinogen is activated into chymotrypsin by trypsin, pepsin, enterokinase, bile salts, and the answer will be trypsin. So this is quite fact based. So I'm just going through. Trypsin converts chymotrypsinogens into chymotrypsin. Remember this, and other proenzymes into active enzymes, and trypsin can also activate trypsinogen. Therefore, once some trypsin is formed, there is an autocatalytic reaction. There is an autocatalytic reaction. Trypsin, the chymotrypsin and elastase act at the interior peptide bond. Trypsin, the chymotrypsin, chymotrypsin and elastase act at the interior peptide bond in the peptide molecules and are called endopeptidase. The powerful protein splitting enzyme of the pancreatic juice are secreted as inactive proenzyme. So inactive proenzyme. First of all, so pancreatic juice are secreted as inactive proenzymes. Then they are activated. Otherwise, they will self-digest. Self-digest from where they are stored, from where they are produced. Okay. So trypsinogen is converted to active enzyme, that is trypsin, which in turn activates chymotrypsinogen into chymotrypsin by enterokinase. Chymotrypsin. Okay, so the trypsinogen converted to active enzyme trypsin by enterokinase when the pancreatic juice enters the duodenum. So overall, pancreatic juice, it enters the duodenum. And trypsinogen then is converted into trypsin by enterokinase, and this trypsin will act on chymotrypsin to form chymotrypsinogen to form chymotrypsin. Okay, so this is the overall story that is happening here. Okay, next, incorrect about bilirubin. So incorrect about bilirubin: conjugation by UDP glucuronide transferase in liver, breakdown of glucuronidase in gut to increase free pool of unconjugated bilirubin normally excreted in urine can cause BBB, blood-brain barrier. So answer is normally excreted in urine. It is obviously not normally excreted in urine. So normally, answer will be normally excreted in urine. So first option is conjugation of UDP glucuronide transferase in liver. This is true. Okay. Bilirubin helps in the conjugation of UDP glucuronide transferase in liver. It helps in breakdown by glucuronidase in gut to increase the free pool of unconjugated bilirubin, and it can cross blood-brain barrier. That's why kernicterus occur. Too much increase of bilirubin in jaundice leads to kernicterus. Okay, especially in the neonatal patients. So that's why. So remember that bilirubin can cross BBB or blood-brain barrier. So anyway, bilirubin excretion in urine is seen in obstructive jaundice. In obstructive jaundice, bilirubin excretion occurs in urine. Otherwise, there should not be bilirubin present in urine. And in obstructive jaundice, what happens? Conjugated bilirubin regurgitates back into the bloodstream and cannot be excreted secondary to obstruction. So this bilirubinuria is seen in obstructive jaundice, leading to mustard yellow discoloration of the urine. So bilirubin in urine indicates pathology. It's abnormal. It's not a normal condition or abnormal condition. It's not normal mechanism. Okay. Anyway, conjugated bilirubin in normal people is broken down by an enzyme by name of beta glucuronidase to form unconjugated bilirubin. This is called enterohepatic cycle. This is called enterohepatic cycle. So conjugated bilirubin in normal people is broken down by the enzyme beta glucuronidase to form unconjugated bilirubin. Okay, so this statement, option number B, is true. Now conjugation of bilirubin occurs in liver. Conjugation of bilirubin by UDP glucuronyl transferase. This is an enzyme, and so conjugation of bilirubin occurs in liver. This is also true, as we have marked. So conjugated bilirubin occurs in liver. In case of defective conjugation like Crigler-Najjar syndrome, the level of unconjugated bilirubin increases severely and it crosses BBB, leading to kernicterus, especially in Crigler-Najjar syndrome. So option number D is also correct. Option number A is correct. Option number B is correct. Which one of these is incorrect? Normally excreted in urine. This is false statement. Somatostatin is secreted by alpha cell, beta cell, gamma, and delta cell. By delta cell. Somatostatin is secreted by delta cell. So you need to know that human pancreas has islets of Langerhans cells. Human pancreas has cells 1 to 2 million of islets of Langerhans. Islets of Langerhans. These islets contain 3 major types of cells, that is alpha, beta, and delta. There is no gamma. Alpha, beta, and delta. 60% of the cells of islets are mainly in the middle of each islet, and that is beta cells. Beta cell constitutes 60% of the islet of Langerhans. 60%, and it secretes mainly insulin and amylin. Insulin and amylin. And alpha cells constitutes about 25% of the total islets secreting glucagon. Alpha cell 25% of the islets and 25% of the islets and secrete glucagon. And delta cells about 10%. About 10% and secretes 10% and secretes that which is given here somatostatin or SS we call it. Okay, so this is about islets of Langerhans. So all of the following are actions of cortisol except increase blood amino acid, decrease liver amino acid, decrease blood amino acid, increase liver amino acid. So the answer will be C, decrease blood amino acid. Decrease blood amino acid. Now you see action of cortisol. Action of cortisol, you need to know that cortisol raises free amino acid in this serum, which free amino acid in the serum. So cortisol what it does, it increases amino acid in the serum or free amino acid. Free amino acid in serum. Okay, and by inhibiting collagen formation, decreasing amino acid uptake by muscle, and inhibiting protein synthesis. So how it increases the amino acid? What are the mechanisms? Number one is inhibiting collagen formation. Inhibiting collagen formation. Then it decreases amino acid uptake by muscle. Amino acid uptake by muscle and inhibiting protein synthesis. Inhibiting protein synthesis. So by these three mechanisms, it increases free amino acid in serum. And glucocorticoid promote protein degradation and inhibit entry of amino acid in tissues, and also lead to utilization of amino acid by promoting gluconeogenesis directly and by permissive effect on growth hormone. So glucocorticoid promote what? Promote protein degradation and inhibit the entry of amino acid in tissues. Inhibit entry of amino acid in tissues and also lead to utilization of amino acid by promoting gluconeogenesis directly and by permissive effect on growth hormone. Okay, so it increases blood amino acid, but it this option decreases blood amino acid. This is wrong. Okay, it increases amino acid, not decreases. Milk ejection. Now next, milk ejection is facilitated by prolactin, oxytocin, growth hormone, ACTH. It will be oxytocin. 2 points about milk ejection. 2 points. Number one, two hormones are related. One is prolactin, another is oxytocin. Very simple. Prolactin is development of female breast. So development of breast plus milk secretion. plus milk secretion, or we can say milk production actually. Okay, milk secretion, rather we can say production. Oxytocin, always remember oxytocin is ejection of everything. Ejection, or we can say contraction of any bag-like structures is done by oxytocin. In both cases, in case of breast, the lobules of the breast are contracted contracted by oxytocin. Okay, contracted by oxytocin, and it leads to ejection of milk plus uterine contraction also. Uterine contraction. So contraction of any bag-like structures, whether it is uterine or lobules of the breast, it is done by oxytocin. Okay, oxytocin, no production, not actions like those. It just bag-like structures, it contracts. It helps in contraction of that bag-like structures. It can be lobules, it can be uterus. So when the lobules are pressed, lobules are contracted, lobules of breast lobules are contracted, what happens? Milk is ejected. So brought about by oxytocin. And PRL makes things. Always remember, in simple language, prolactin, PRL means prolactin. It makes, it manufactures, helps in manufactures or development or manufactures, development of breast, manufacturing milk. Okay, that is prolactin. And oxytocin, extraction or ejection. Okay, this is oxytocin. So milk production, prolactin; milk ejection, oxytocin. Here they asked for ejection, so answer is oxytocin. Okay, so next we have so many questions. We will go through those questions because there are many questions actually. There are many questions, there is no end to it, but we need to cover overall, give a good coverage of this subject. So anyway, we will start with one by one and we will see what are the answers. So the first question, you see, true statement about melatonin are all except. The true statement about melatonin are all except secreted by pituitary gland, secreted by pineal gland, associated with sleep mechanism, can be used to treat jet lag. Can be used to treat jet lag. So the answer will be true statement about melatonin are all except. So which one of these is false? Melatonin is secreted by pineal gland, as we know. It's very easy actually, secreted by pineal gland. Its synthesis, that means melatonin synthesis and secretion is increased remarkably in the dark period of the day. Okay, that means night time, and maintain at low levels in the during the daytime. And it is associated, as you know, melatonin is associated with sleep mechanism. Sleep. Melatonin is associated with sleep mechanism, and melatonin acts as inhibitor of gonadotropic hormone secretion, and gonads become inhibited and even partly involuted. So it also acts as inhibitor of gonadotropic hormone secretion. Okay, so the main thing is Which one of this false statement? Secreted by pituitary gland. Number A will be the answer. Okay, so the melatonin is secreted by pineal gland. True. Associated with sleep mechanism, that is also true, and can be used to treat jet lag. That is also true. Because used to treat jet lag because melatonin nowadays is used to manufacture certain drugs that can be used to treat jet lag and insomnia in elderly people. Okay, that is why melatonin can be used to treat jet lag. That is also true. Next question, separation of first polar body occurs at the time of ovulation, menstruation, fertilization, implantation, and the answer is ovulation. Separation of first polar body occurs at the time of ovulation. A will be the answer. So, as you know, ovum, right before it is released from the ovarian follicle, its nucleus divides by meiosis, and the first polar body is expelled from the nucleus of the oocyte. So right before the ovum is released from the ovarian follicle, its nucleus divides by meiosis, and first polar body comes. Okay, so the first polar body ovum divides into first polar body, and then ovulation occurs. So the separation of first polar body occurs during ovulation, and the primary oocyte becomes secondary oocyte, right? So the primary oocyte divides by meiosis before its expulsion. Okay, before its expulsion from the ovary. This is about ovum. First oocyte becomes secondary oocyte. Secondary oocyte and first polar body. Okay, then this secondary oocyte, by ovulation it get expels. So this first polar body occurs at ovulation. Okay, next, sperm head contains obviously nucleus. Sperm head contains obviously nucleus. Next, Sertoli cell secretes which hormone? Sertoli cells secretes which hormone? So before going to the answer, first we see Sertoli cells. First of all, Sertoli cells are found in seminiferous tubules. So seminiferous tubules, you have to understand that seminiferous tubules is lined by primitive germ cell and Sertoli cells. Primitive germ cell and Sertoli cells. Now maturation of spermatozoa takes place in the cytoplasm of Sertoli cells. So inside the Sertoli cell, maturation of spermatozoa takes place. Okay, and mature spermatozoa and then release from Sertoli cells and become free in the lumen of the tubule. Which tubule? Seminiferous tubule. Okay, so if this seminiferous tubule is like, is a like tubule, like a tunnel, like a tube. That's why tubule it is named as tube. It is called as tubule. In this tubule, the lining cells are composed of germ cell, primitive germ cell, and Sertoli cells. And inside Sertoli cells, spermatozoa matures and get into the lumen. Mature spermatozoa are kept in are released into the lumen of the seminiferous tubules. So next is, let's Sertoli cells secrete androgen binding protein, inhibin, and androgen binding protein and inhibin. So not testosterone, obviously, because testosterone, as you know, testosterone is secreted by which cell? Leydig cell. And LH and FSH. LH is secreted by anterior pituitary gland. Both LH and FSH are secreted by anterior pituitary gland. Both are secreted by anterior pituitary gland. Okay, and inhibin is secreted by Sertoli cells. Other hormone that Sertoli cells secretes is ABP, or androgen binding protein. Okay. So all are true about capacitation except changes that occur in spermatozoa. So in this option will be B. Okay, this inhibin. Okay, next question. All are true about capacitation except changes that occur in sperm to penetrate ova, occurs in female genital tract, occurs in male genital tract. Sperm loses its cholesterol content in the process. Okay, so the answer will be occurs in male genital tract. Because see, about capacitation. What is capacitation? First of all, we need to know. Capacitation is the process that makes spermatozoa to undergo physical change to fertilize an egg in female genital tract. That means capacitation is nothing but the process of maturation of spermatozoa in order to make it capable of fertilizing an egg or the ova. Okay, that is capacitation. So it occurs in male genital tract? No, it occurs in female genital tract. This is correct. It occurs in female genital tract. So spermatozoa in the male genital tract are exposed to floating vesicle from seminiferous tubule. As we just discussed, in seminiferous tubule, the matured spermatozoa will be kept. This spermatozoa will be kept in seminiferous tubule, and after ejaculation, the sperm get deposited into the female vagina, which swim away from cholesterol vesicle upward into uterine cavity. Okay, and lose excess cholesterol, and membrane of the head of the sperm becomes weaker, which makes penetration in ova easy. So anyway, the option A, changes that occur in sperm to penetrate ova. That is only the definition of capacitation. It occurs in female genital tract. Obviously, it occurs in a female genital. It loses its cholesterol content in the process. Because in seminiferous tubules, what happens? Spermatozoa are exposed to vesicles. Spermatozoa that are present in seminiferous mature spermatozoa after coming from Sertoli cell, they are present in the lumen of the seminiferous tubule. Okay. Now they are exposed to some vesicle. Vesicles containing what? Containing cholesterol. Vesicles with cholesterol. Okay, that cover up the sperm acrosome and makes the membrane tough. So what is the function of this cholesterol? It protects the spermatozoa. It keeps the cover of the spermatozoa or membrane of the spermatozoa tough enough, so that it doesn't get broken down in between. Then when it is ejaculated into vagina, it goes to the vagina and swims upward into the uterine cavity. After reaching the uterine cavity, it loses excess cholesterol and membrane of the sperm head becomes weaker, because there is excess, this cholesterol get lost in the uterine cavity, in the uterus. Okay, and the head of the sperm becomes weaker, because this cholesterol was responsible for protecting the head of the spermatozoa. Now this cholesterol have been shed, so there is no one to protect the sperm head, and this sperm head, what will happen, will become weaker and makes penetration of ova, and can penetrate the ova very easily. Okay, so this is the number D, explanation of the number D. So this is also true, but obviously this number C is not true. Okay, next question. Defect in leptin leads to